Lacrosse and actual sport, yeah. It's dumb. You know what is a sport?
Okay, that clock back there is getting slower and slower every day. It's actually 1:30, so try to remember that's slow. Okay, so evals until this Sunday. I'd appreciate if you do those. Constructive criticism is always better than outright name calling, right? But if you especially let me know what you think about the book, OpenStax, and the homework. So if you find that helpful, if you don't find it helpful, like that I'm especially interested in that. Okay, I'm putting the finishing touches on the exam. Once I have that done, then I will post some practice questions for you. In addition, I will post this question. I just thought I'd show this up here. I don't know if many of you have had a chance to read it yet, but I got this from a test bank for medical students take this step 1 exam in their 3rd year of medical school. So it's full of questions that are all like this. This was from the infectious disease portion of it. Again, a practice question. And I thought that some of you may be able to take a crack at this. They give you a lot of information that you don't really need. But if you look at this, a large gram-positive rod is causing this infection. Shortness of breath, there's a big clue. And this is another thing that we talked about, widening of the mediastinum. Okay, so from that information, you might guess that this patient has, not TB, that's a good guess, anthrax. So gram-positive rod, TB is, and sometimes that is considered gram-positive, but okay. And then, so that's the first step, understanding the pathogen, and then it asks you what virulence factors have made it successful. And remember, anthrax, Bacillus anthracis had that kind of capsule that has that a little bit more protein composition. So, and just kind of more challenging question to show to you. And again, I'll post that with some other practice questions here, hopefully in the next couple of days. All right, so where we left off last time, I think we're in pretty good shape. I think we're gonna finish okay. If we have a little extra time, we'll do some practice questions in class on Wednesday as well. I think it's gonna come out about right though, in terms of getting through everything that I had anticipated that we would. So go back, I did post some learning objectives where I had crossed off things that we're not going to get to. So that's it's like a revised learning objectives on Blackboard. Okay, so we finished Neisseria gonorrhoeae, and we started its cousin, Neisseria meningitidis. So these two organisms share a lot of the same virulence factors, very, very different kinds of disease manifestation though. In this case, Neisseria gonorrhoeae, not normal microbiota ever, as far as I know. Interestingly, even though the Neisseria meningitidis causes a very severe infection, some of us could be carrying it around with us right now, and it's just in really low numbers. It's more of a not something that we would carry our whole life, just kind of maybe transiently normal microbiota in our upper respiratory tract, and we can pass that to somebody else through those large droplets. Most of the outbreaks on college campuses have usually been able to be traced back to some party where a bunch of people were sharing drinking glasses. So this causes meningococcal meningitis. There are actually, we talked about a couple of other organisms, bacteria, that cause meningitis. There are other kinds of meningitis, like there's viral meningitis. I mentioned that. That's usually not as serious because they don't get the toxin production from viruses. There's also fungal meningitis. There's also what they call parasitic meningitis and amoebic meningitis, like all different things can cause this inflammation of the meninges. And it manifests, you know, a little bit differently, but and the fungal and the parasitic is pretty rare, actually. There's also a non-infectious meningitis that can be caused by cancer, so among other things. So, okay, early symptoms. What are some of the early symptoms of meningococcal meningitis? Anybody throw it out? What's it most famous? What's the one that you kind of think of first? Yes, stiff neck. All right, so that's stiff neck and headache. Not just a little headache, but the worst headache you've ever had in your life. All right, so you know, you can get kind of a kink in your neck if you sleep wrong, and maybe if you have a fever or something, then you might worry about it, but that's got to be accompanied by, again, like the worst headache that you've ever had. Other things like fever, sore throat, nausea, sometimes an increased sensitivity to light, which is maybe a symptom that you don't often see. Okay, and so most reliably, you know, a patient that has those symptoms should go to the med center or the ER right away, because this is an infection that can move very quickly. To definitively diagnose meningitis, they'll have to do a spinal tap. They'll have to take a look at the spinal fluid and take a look at it. So it's kind of invasive to know for sure, but if you know a patient fits all the right symptoms, they'll order that. Those are some of the early symptoms. The infection, like I said, will very quickly progress. This is a gram-negative pathogen. Remember, it's a cocci, gram-negative pathogen, and all gram-negatives have endotoxin, which is actually lipid A, which is part of the lipopolysaccharide of the cell wall, that outer membrane of the cell wall. Normally, that lipid A is shed maybe to a small amount whenever the bacteria divides and that outer envelope is disrupted, more so when the bacteria dies and the cell wall starts to break down. But Neisseria meningitidis actually is releasing that outer membrane all the time, and we call that process blebbing. Okay, so releasing that endotoxin all the time. So we're getting, 100, let me write that again, 100 to 1,000 times more endotoxin than normal. And it's more toxic than normal. Okay, so that endotoxin release in massive amounts is causing some of these later really particularly serious symptoms like petechiae. Petechiae it's a rash that kind of starts out maybe a deep red, it turns a little purple, and then it turns black. So we're getting some inappropriate blood clumping, agglutination, some capillaries breaking because of that. So these are black spots. It's kind of like a black rash. So you can see that here. Those petechiae will kind of coalesce into, you know, larger areas of black color change on the skin. When you see that black, you think necrotic tissue. That's death of tissue.
I was just about to ask, is it necrotic?
Yes. Yep. So this baby's hand, that would have to be amputated. Yeah. So patients can go into shock. All right. Remember, that's main thing. The endotoxin can cause inflammation. Too much inflammation, too much leaky blood vessels causes the blood pressure to go way down. Patients can go into shock. Extremely high fever, neurological complications with that, cardiac arrest. this has, what's the mortality rate, the kind of ongoing hot spots, meningococcal meningitis right now is taking place in Florida. And they're saying with that outbreak, about 25% mortality associated with that. So pretty high mortality. And the people that do survive, maybe 20% of them are left with some permanent disability, like mental retardation or blindness or deafness or loss of limb due to amputation. So pretty serious outcomes. Who's at risk? This is very rare. Who's at risk? Anybody can get it, but if very young children, especially infants less than 1, are more susceptible to many different things, not just this, but the capsule here is actually a T-independent antigen. So remember we said that young children don't have a very mature B cells, so they don't have very good response to T-independent antigens. Kind of the next age group is young adults. Again, college campuses have been the source of lots of outbreaks just because they're kind of living communally, perhaps. It is rare, and if somebody maybe would get meningococcal meningitis a second time, then they start to think, look at any immunological deficiencies, and it turns out individuals that have some kind of complement deficiency are more susceptible to this infection, especially individuals without spleens are more susceptible to a lot of bacterial infections, and then kind of anybody that's immunocompromised, that's kind of a catch-all. Interestingly enough, incidence of meningococcal meningitis is a lot higher. They call it the meningitis belt, kind of an area of sub-Saharan Africa. We don't totally understand why incidence is higher there. It may have something to do with just kind of a lower standard of living, malnutrition, also something to do with the weather, because cases are highest in their dry season. So kind of hot, dry climate, compound with, again, nutritional deficiencies may have something to do with that. All right, so I just pasted a couple of charts from the CDC. Again, young children, it's pretty high. We have this kind of bump, young adulthood, and then it goes up again. Older adults, they're just, their immune response is not as robust as we age. Over time, how are we doing? Is this getting better? It's getting worse. It's definitely getting better. We had our first vaccine come out about 1978. So maybe once that gained in popularity, incidence is going down. And then we came out with a better vaccine in 2004, which is slightly different from the vaccine that we're using now even. So the best I can see is that we have around 300 cases a year in the US. Like I said, right now, there's an outbreak in Florida that started a little over a year ago, like the end of 2021, and is still ongoing among men that have sex with men. This is not a sexually transmitted infection, but it does take close contact. And so that's sort of just happens to be the population of people that it's kind of circulating, and they're really trying to get vaccinated. I think there was like some 60 some cases in that outbreak so far. All right, so thinking about virulence factors, a lot of the same ones as Neisseria, gonorrhea. Remember, one of the big things that we talked about with the gonorrhea is the antigenic variation, how it can change its proteins in its papilli or fimbriae very quickly. Neisseria meningitis is not as good at that. Okay, so it's not as variable. Like once we make antibodies, they do work for it. So it doesn't quite have that same trick, but it has the same kind of variation of the lipopolysaccharide, oligosaccharide, which again, an endotoxin is included in that. Remember, IgA is the main antibody on our mucous membrane, so it has this enzyme that can kind of degrade our antibodies. And the fimbriae and the pili that are specific, not this time for the genital tract, but specific for the meninges. And that's kind of those layers of membrane that surround our brain and spinal cord. Okay, unlike Neisseria gonorrhoeae, this organism does make a capsule. Again, the main function of that is going to be anti-phagocytic. Okay, keeps our complement, even our complement has a hard time adhering to this organism. It's just not, okay, so again, outwitting our white blood cells. There are 5 main serogroups of that capsule. We think that there's kind of 12 variations of that capsule in all, but they name those serogroups by letters. So ABC, your book includes X, so I'll write that in here, Y and W. So I guess that's 6 and not 5. But those seem to be the most common serogroups of the capsule. That the latest outbreak is been that C serogroup in Florida. Okay, so all right. And then the most recent one in college campuses have been that B serogroup. There was about 10 years ago at Princeton, about 13 students had an outbreak with that B serogroup. Okay, so hold that thought. Those serogroups are important when we think about the vaccine and how we formulate the vaccine. What do we do to treat this? Oftentimes, cephalosporin is one of the antibiotics used. There could be other ones. I haven't updated that lately, but kind of a later generation one that is a little bit more broad spectrum, a little bit better at crossing that blood-brain barrier. Also, anti-inflammatories to try to counteract that endotoxin. Not just Tylenol, but things like steroids, kind of tamp down that inflammation. Anybody that may have been exposed, they'll give antibiotics to prophylactically. So that means before you show any symptoms at all, just in case you may have been exposed just due to the seriousness of the infection. The vaccine. So they just changed the name of this a few years ago, but like I have pictured here, it's called Neisseria. All right, so they call quadrivalent. It covers the 4 of those capsule types. Why not X? I'm not really sure. They have a separate vaccine for that subgroup B. For whatever reason, I don't know, they couldn't include it there in one. If every once in a while an ad will pop up on my feed somewhere about the vaccine for meningococcal B. I forget if they have a special name for it, but it's kind of controversial because it's so rare. Do you widespread vaccinate people for something that they're very likely to come in contact with? Some people argue yes, because again, it can be deadly if you contract it, but some people argue it's not worth the cost and possibility of, having reactions to the vaccine. But this Neisseria, that is recommended. Usually people will get the first dose around age 10 and 11 and a booster at 16, and that's going to protect against the most common kinds. What kind of vaccine is it? It's we call it a meningococcal conjugate. Okay, so it's got, parts of the capsule, and those weakly antigenic, polysaccharides from that capsule are kind of somehow attached to some protein so that our immune system notices those capsular antigens better. I don't know what protein that it is. Okay, if babies are at high risk for this, why don't we give the vaccine to babies? Because it doesn't do any good. Okay, they don't form memory cells to it, and so we have to wait till kids are a lot older. All right, questions? Any questions about meningococcal meningitis? I think I was trying to sometimes, before lecture, I think of questions that I haven't thought of before, and I was trying to understand if meningitis looks the same, whether it's from, strep pneumoniae or Haemophilus influenzae. I think the petechiae is unique to the meningococcal meningitis, but they do share a lot of the same symptoms. All right, so I want to flip now and start talking about some viral pathogens. We'll talk about some DNA viruses today, and then finish next time with some RNA viruses. See how far we get. Remember, I did have written into the objectives that we would do whooping cough, but we're gonna skip over pertussis. All right, so just a few kind of reminders of what viruses are. Sometimes we study the details and we forget the big picture of things. So remember, viruses, they can only reproduce inside of a living cell. Right? So we can't grow them in the lab in a broth culture, right? We just can't give it some nutrients and expect them to reproduce. The virions, we call them virions when they're outside of a cell, and then more correctly call them viruses when they're inside of a host cell. But they're composed of nucleic acid, either DNA or RNA, not both, one or the other, as well as a protein capsid. Okay, so a capsid made of these proteins, capsomeres, that are going to protect that nucleic acid. Oftentimes, viruses that infect animals, humans, are going to be enveloped as well. Much more rarely bacteriophage. It's much more rare for them to be enveloped. There are a few exceptions there, but sometimes as well, they have that phospholipid bilayer. Okay, and I just like this little graphic over here. We talked about how we classify viruses. Sometimes we try to put them in families and orders and genera, just like we do living organisms. So it's got some of those families listed there. It also has these split up into their Baltimore classes, which remember is based on what kind of nucleic acid they have, whether they're double-stranded DNA or single-stranded DNA, for example. Okay, so not to memorize, just to kind of, remind us kind of what's out there and show some different capsid shapes and things like that. All right, so really two different DNA viruses I want to spend some time on. Herpesvirus is a good example of a double-stranded DNA virus. You'll find some homework questions here in 22.4 in your textbook. This is kind of an old graphic here that calls these viruses herpes simplex viruses. Although your textbook does use that term herpes simplex, I understand more likely we call them human herpes virus now rather than herpes simplex, but it's the same thing. Okay, so notable characteristics. Like I said, they're enveloped. It's a big family of viruses, but they all have these characteristics in common. They have an icosahedral capsid. So remember, that's 20-sided. Most often, those equilateral triangles, high strength and stability with a icosahedral capsid. of polyhedral capsid, and linear double-stranded DNA. Okay, so like in the picture here, you can see it's all one strand of DNA. It's linear is the same thing as saying unsegmented. Okay, so all in one line. In addition, the herpes viridae all have something that's not unique to herpes, but it is very pronounced and kind of one of the things that we look for that makes this virus unique. It has a bunch of proteins that we refer to as the tegument proteins. So in this picture, it's all the little kind of purple circles on the inside of the envelope. Those are the tegument proteins. We think there's somewhere maybe like 20 different proteins that the virion carries with it. Proteins that are going to suppress host immunity and aid in viral replication. So an example of suppressing host immunity, there are proteins that it carries with it that will down-regulate the host cell MHC1 presentation. All right, so that's a really dirty trick that they do. That's the mechanism we have, remember, for all our cells to advertise what's going on inside our cells. That's the way our cytotoxic T cells and our helper T cells will notice that one of our host cells is infected. All right, so if we're not able to do that, then the virus is more likely going to live quite happily inside of our host cells. Another protein will actually inhibit the production of type 1 interferons. So that's interferon alpha and beta. Okay, and remember that's one mechanism, kind of a non-specific mechanism that our cells have to kind of slow viral replication. It's they secrete it, and then the cells around them can kind of prepare this inactive antiviral protein. So two ways that helps that's two examples of the way those tegument proteins help the virus be successful. Lots of glycoprotein spikes, and we won't get into too much detail here like we will with influenza, but those are necessary to bind to certain host cells and enable the virion to fuse with the host cell membrane. We think members of this family of virus are extremely widespread. Probably almost 100% of us have been infected at some point with a herpes virus. In some cases, our immune system fights it off and it doesn't go latent. But I'll get to that in a minute. All members have the ability to show latency. So remember, that's the lysogenic cycle that we talked about in bacteriophage. In animal cells, we call that latency. Let me back up a minute. Reservoir, pretty much humans. There are herpes viruses in animals. They typically don't cross the species barrier. There's one exception. It's human herpes virus. I think they call it human herpes virus 9. They've been, in rare instances, that has crossed a species of monkeys. Humans have got herpes virus from a species of monkey. There's been about 50 cases of that, and about 80% of those people have died. So it's very rare, but it's very deadly. That one exception has, again, the rare instance that it's zoonotic. Okay. So latency. Interestingly enough, with bacteriophage, that's in lysogeny, that virus will always form a prophage. So it'll always incorporate into the bacterial chromosome. Herpes virus actually doesn't incorporate into the chromosome. It actually forms these structures called episomes, which are plasmid-like circles of DNA. Okay, so if here's our host cell, and that's a really simplification because a lot of times the host cell would be a nerve cell. Here's the nucleus of the host cell, and here's our chromosomes. An episome would be just like a little circle of viral DNA that's kind of associated or near our chromosomes, but not actually part of it. And every time that cell replicates, that viral DNA, that episome is going to replicate as well. Okay, so permanent. Even though it's not right in that chromosome, it gets replicated every time. And as far as we know, there's not any way to get rid of it once it's there. So in other words, there's no induction. That's what we call it when a lysogenic bacteriophage will enter into the lytic cycle and blast out of the host cell. Some herpes virus are oncogenic. There's been a couple of them that cause cancers. This is a really nice 2-minute YouTube video that's really helpful. It's just a drawn animation understanding how a herpes virus will infect a cell. And it goes in a little bit more detail than what I want to here, but it's a nice kind of visualization of how it infects a host cell and how it leaves again. All right, before we get into the specific disease, a little bit more general characteristics of lots of different herpes virus disease. They target mucosal membranes. Okay, so basically any little tiny cut that we have that will enable the virus to gain access. You think around our lips and our mouth, when the weather's dry, especially, we might have a little crack in our lip, and that's how we can get cold sores real easily if we're exposed to it. They're going to multiply in the basal and epithelial cells. So the epithelial cells are kind of the layer of cells that lines all of our body parts, including our veins, our blood vessels. And then basal cells are kind of the lower layer of cells right below that. They cause a lot of inflammation in those cells. Usually localized in that particular reason, not system-wide inflammation, and causes sometimes death of those infected cells. Sometimes those cells will actually fuse, the infected cells will fuse with other cells that are not infected. So when we're looking to diagnose herpes disease, we don't very often take a tissue sample and then look for viruses under the electron microscope. It's just really inefficient and expensive to try to do that. Oftentimes what we do instead is look for a cytopathic effect, and sometimes we just call this CPE. A cytopathic effect, that means viral changes in host cells that are visible with light microscopy. Okay, so for instance, we didn't talk about rabies this semester, but when we're diagnosing someone that's been infected or an animal that's been infected with rabies, we took a sample of brain tissue and looked for these little dark spots in neural cells called Negri bodies. It's a different cytopathic effect that we look for herpes virus. We look for something called syncytia, which means multi-nucleated, I ran out of room there, multi-nucleated cells. Okay, we look for cells that have more than one nucleus. So you can see in this example here, all these little dark spots in this nerve cell, those are nuclei. Okay, this is, and over here in this picture, again, all these dark spots, I don't know what kind of cell this is, not a nerve cell. So this is a really, like I said, kind of a dirty trick that the herpes virus does. Instead of moving from one cell and infecting that, and then the new virions going out and infecting other cells, when those virions are released from the infected cell, they're going to be a lot more susceptible to maybe our circulating antibodies that would bind to those virions and keep them from adhering to new cells. So what the virions will do instead is cause our host cell to fuse with another host cell that hasn't been infected. And in that way, those virions are not susceptible to being noticed by our immune system. So you get these cells that have been fused with, over and over and over again. And so that's why they have lots of nuclei. So herpes virus is not the only infection that causes a cytopathic effect, but put that together with the symptoms of the patient, and then that's a diagnosis. All right, let's talk about the different, I guess these are most correctly called genera, maybe, of human herpes virus. There's actually 9 different human herpes virus, well, 8 or 9 if you count that one that's in monkeys. HHV1, or sometimes it's called human simplex virus instead of human herpes virus. That's a causative agent of fever blisters or cold sores. And HHV2 are genital sores. Okay, so sometimes people refer to HHV1 as above the waist herpes and HHV2 as below the waist herpes. That doesn't mean that they can't, cross. Like people can get HHV2 in their mouth. It's typically when people are doing non-traditional sexual practices. Okay, HHV3, we'll talk about those in a little bit more detail, as well as HHV3. Anybody know what varicella zoster virus causes? Yes, chickenpox. Okay, so we'll talk more about that. HHV4, sometimes I talk about that. I'm going to leave that off this year just to kind of move on to other things. But Epstein-Barr virus, some of you have probably had that. That causes infectious mononucleosis. And it can actually cause a lot of different things. That's one of the oncogenic herpes virus. It can cause a B-cell lymphoma called Burkitt lymphoma as well. It's pretty rare, and some other things. So, all right. So let's, and then there's, the other ones, some of the HHV6 and 7 cause some kind of childhood infections like I think roseola is actually herpes virus. HHV8 causes Kaposi's sarcoma, which is basically it's only people that are totally immune compromised, like AIDS patients that get. Okay, so there's some other ones out here, but we'll focus on the first 3. All right. All right, HHV1. The technical term for cold sores is gingivostomatitis. And these are kind of painful vesicles on the lips or maybe just inside the mouth. Canker sores, though, if you've ever had canker sores, that is not caused by herpes. That can have a lot of different causes, but not herpes. So usually the herpetic cold sores, again, lips, maybe just inside the lips. How are they transmitted? Close contact. Okay, so usually, kissing or sharing drinking glasses or maybe sharing eating utensils. They usually last maybe 7 to 10 days. Maybe the day or so before they erupt, the patient might feel some kind of tingling or itching, kind of a prodromal phase there. And actually that HHV1 can manifest in other areas besides the lips. People can get them on their fingers. Most of us very easily would have, little cuts on our cuticle where they can gain access. This is the main reason that dentists and dental hygienists wear gloves, so they don't get herpes infection on their fingers. That's also called Whitlow's disease, or maybe just Whitlow. And we can also get herpes lesions on our eyes. So especially if you would have those lesions on your finger and maybe you rub your eye, or I don't know, rub your mouth and your eye. That's the technical name for that would be herpetic keratosis or keratitis. That can make patients really sensitive to the light, has the potential to cause some permanent vision damage. So, okay. And then in really, really rare cases, people can get a herpetic infection in the brain or the spine. We would call that herpes simplex encephalitis. I think encephalitis is an inflammation of the brain. That's thankfully very rare because it's also very deadly. So that's usually people that are, again, really immunocompromised, like for instance, an AIDS patient. Okay, substantial mortality associated with that. All right, and I spared you the graphic pictures this time of the genital herpes. Targets mucosal membranes in the genital tract, of course, lesions on the genitalia. We said that gonorrhea was really, really treatable. Of course, you can get it over and over again, but at least, again, it's treatable with antibiotics. It's estimated that 1 out of 6 adults have HHV2, have genital herpes. And that's sobering because once you have it, we don't have a drug to get rid of it. We have drugs that can make it, have outbreaks less commonly and not as severe, but once you have it, at least for now, you have it. And people can spread this to sexual partners even when there are not actual active lesions, more commonly when there are active lesions, but there can be some viral shedding, of a few cells here and there, and it can possibly be spread anytime. That can cause itching, discharge, sometimes fever. And on average, patients will have maybe 2 to 3 outbreaks a year. Can be much more common than that, but kind of that's the average number that I've seen. Young babies can get neonatal herpes during birth. So for that reason, mothers having an outbreak, they'll deliver the baby by cesarean section. Maybe even whether the mother's having an outbreak or not, they'll deliver the baby by C-section. Babies can contract an eye infection that looks a lot like that neonatal ophthalmolum that we talked about for gonorrhea. Depending on how well the baby's immune system responds to that, it can actually spread to the central nervous system and cause substantial mortality. So a little bit more serious infection. Okay. In addition, mothers that are infected have a little bit higher incidence of miscarriage and preterm delivery. All right, so we talked about how all the herpes viruses have the ability to go latent. So let's talk about latency in HHV1 and HHV2. Someone with that HHV1 that has a fever blister, that will typically kind of travel down one of the branches of the cranial nerve number 5. It's also known as the trigeminal nerve, okay? Cranial nerve number 5. That's typically where fever blisters would go latent. There's several branches of that, the ones in the jaw, the maxillary and the mandibular. And if a patient has, they call it ocular herpes here or hepatic keratitis, it would travel up the ophthalmic branch. Okay, so again, HHV1 goes latent in the trigeminal nerve. Whitlow would more likely go latent in the brachial ganglia, and then HHV2 would likely go latent in the sacral ganglia. Okay, so in nerves, when does a patient experience reactivation? Typically in times when they're under some kind of stress. And it might be things that you wouldn't normally think about as being stressful, like patients in the sun a little bit too much. So maybe a sunburn or just out in the sun when they're not used to it. Menstruation. Okay, so that could be considered a stress. Some kind of immunosuppression. So maybe the patient has to go on some prednisone, some kind of steroid drug for some other condition. Or maybe they just, are ill with the flu or something and have the fever, have some kind of fever. Those are common stressors that can reactivate some herpes sores. My husband got a cold sore for the first time in 20 years after he got the second COVID shot. So that was interesting. Like that stressed his body enough that that came out. The good news is usually, after the first time that the blisters emerge, usually when they're reactivated, it's not quite as bad. Our body does mount some antibody response, and usually it's not quite as bad when it does reactivate. Okay, symptoms are a little bit less severe. So what do we do about this? Like I said, we don't have a cure. The drug acyclovir is really the best one that we have, and they talk about some different kind of permutations of acyclovir in your textbook. But this is a nucleoside analog. So we talked about these a while ago. Remember, something that's a nucleoside analog is going to mimic the action of a nucleoside. And a nucleoside is going to be the base and the deoxyribose. Okay, so here's the structure of acyclovir. Here's a guanine. It's the same base as the regular nucleoside guanine, but then you can see the deoxyribose looks quite a bit different than real deoxyribose, but the DNA polymerase apparently that the herpes viruses use doesn't really care. It's kind of indiscriminate in what it throws in there when it's replicating its DNA. So we'll incorporate acyclovir, but there's no 3' hydroxyl group there. So it terminates the chain, and the virus can't add any more nucleotides onto that. So it halts viral replication. So hopefully that would reduce the length of the outbreak. It can be given in pill form. It can be injected. It's in lots of different forms there. In terms of cold sores, there's some over-the-counter stuff like lip balm with like menthol or camphor in it to try to reduce the irritation. There's one other medication called Abreva, which is supposed to, whoops, sorry about that, which is supposed to keep the virus from fusing with the host cell membrane. And that's over the counter as well. It says it might lessen the outbreak of a cold sore by about a day. So a little bit of help perhaps there. And that's, again, over the counter, not prescription. Okay, questions? Let's move on to HV3, varicella zoster virus. It's kind of a complicated name. Varicella means speckled. Okay, I think it's either Latin or Greek, I forget which. And zoster, I think it's Latin for belt. Okay, so that doesn't make a lot of sense when we think about the infection chickenpox, but hold on to that thought. We'll come back to that. Chickenpox, again, write that up here. That's one of the things that this virus causes. Oh, let me back up a second. Somebody see my notes and I forget something. In terms of vaccine for HHV1 and HHV2, there has been one in the works like for years and years and years. And every time I check on it, it says, oh, it's still in clinical trials. It's still in clinical trials. But they're trying to formulate a vaccine to give to people once they have herpes. So not to keep you from getting it, but once a patient has it, a vaccine that will, again, shorten the lengths of the outbreaks and make you have outbreaks less often. They're trying to, it's also in works an RNA vaccine for it. So again, there are kind of treatments, but nothing's FDA approved yet. Okay, try to reduce the viral shedding. All right, so on to HHV3, humans only. With a name like chickenpox, you might think we get it from chickens, but that's not the case. I think it was named that because the rash kind of resembles a chicken once it's been plucked. Perhaps, I don't know. But this is something that only humans get. And we have to be pretty close to each other to pass it on. So respiratory droplets, again, closer than 1 meter, not airborne. And it is, oh, I think that's what I meant to write right there. An interesting thing, it's contagious about 1 to 2 days before the rash comes out. And kids don't typically have symptoms. They don't start to feel bad until the rash erupts. Okay, so it's contagious in the incubation period. So that is the recipe for an infection that's really contagious, because people are walking around with the infection and they don't know they're sick. So they're in contact with people that, you know, they're normally in contact with. They're not sitting home in bed when they're contagious. So when you think about how contagious something is, we can do a calculation that's known as R naught. Okay, so we pronounce this R naught. And this is basically what we would call a reproductive, basic reproductive number. It's a term that we would apply to help us understand how contagious something is. Okay, so it's an approximation. If everybody in the population was susceptible to an infection and you had one person that was sick with it, how many people would they most likely get sick? Okay, how many other people would catch the infections from them? Okay, so if you kind of compare the R naught of chickenpox to some other infections, influenza is probably somewhere an R naught of about 1 to 2. So one person sick with influenza, everybody else is susceptible. You're probably likely gonna just come in contact with 1 to 2 other people that would get sick from you. If the R naught is below 1, then that's an infection that would, at least an outbreak that would die away very quickly. All right, on the other end of the spectrum here, 18 and R naught of 18, that's measles. Okay. That's why we have to keep our vaccination up, rates up for measles, because if it does, show up, lots of people would likely be ill with it. Okay, so compare those two. Chicken pox is about 10. So 10 other people from one person. That's why before we had vaccination to chicken pox, almost everybody just got it when they were a kid. If we compare that to COVID, the original kind of alpha COVID was estimated to be between 2 and 3 other people. Some other variants, I've heard widely different approximation of what Omicron would be, anywhere from like 7 to 15. Depends on who's doing the calculation, and it's complicated by the fact that some people are very different degrees of partially vaccinated. So they had 1 shot or 2 shots or 3 shots or 4 shots, and then so not everybody is susceptible. So it's a complicated calculation to read, and you read different things from that. So, okay. So again, very infectious before kids. Adults tend to have a little bit of a prodromal period before they get the rash, and so they might be not as likely to pass it on. What does symptoms look like of chickenpox? Because I assume that probably not a lot of you have had this. Maybe a few of you. The rash is going to begin on the trunk region and spread from there. Okay. They're kind of small fluid-filled vesicles. They're very itchy. And they usually will persist for maybe 5 to 7 days, even until they start to scab over. And once they start to scab over, that's when they get really itchy. Okay. Accompanying that rash, and usually not until the rash starts, again, things like fever, malaise, loss of appetite. Okay, I can remember having the rash inside of my mouth and in my throat. So I can just remember being totally just miserable with this. And when the vaccine came out, I was like, yep, my kids are getting this vaccine right away. But okay, but usually it clears up on its own. Complications are pretty rare with it. People have a good memory response to it. So most of the time you get chickenpox once, and then you have such good memory cells that you rarely get it again. Sometimes you would hear, oh, somebody had a really mild case, and then they might get it again. But so, for that reason, when the vaccine came out in 1995, there was some controversy there, because people said, this is not something that usually causes a big problem. Why do we want to, risk a vaccine? Well, three reasons why it might be a good idea to vaccinate, and I think the arguments get stronger and stronger as we go. Okay, so one, there is a chance of getting some secondary infections from chickenpox. Most commonly, some kind of staph or maybe a streptococcal skin infection. Right? It's, because it's itchy, we tend to itch scabs before they're ready to come off. And they might bleed a little bit, and that, possibility of getting some skin infection there. More rarely, things like pneumonia or encephalitis, inflammation of the brain. Okay, so there is a chance of having, some complications with, childhood chickenpox. A stronger reason to vaccinate is, remember, it's a herpes virus, so it goes latent. And in this case, in nerve cells. One of those branches, the trigeminal, brachial, or sacral ganglia. And when it does reactivate, and it probably about 1/3 of the people that had chickenpox as kids, it will reactivate again about 1/3 of the time when they're adults. let's see, will reactivate not as chickenpox, but as a rash known as shingles. Okay, and shingles is not an itchy rash, it is a painful rash. So sometimes this is kind of misdiagnosed as other things. Some people might think that they've got poison ivy or something, but it's not, again, it's not itchy, it just hurts. Okay, so here's an example. Most commonly, this will reactivate on the waist region. That's where that name zoster belt, speckled, varicella zoster speckled belt shows up, on one side of the torso or the other. And again, this maybe lasts somewhere between 1 and 2 weeks. It's very painful. Usually it clears up. Sometimes it can reactivate in the trigeminal nerve, and then you get it on one side of the face or the other. It's very striking how it's one-sided because it only reactivates on one side of the nerve or the other side. Usually it clears up without too much trouble. About 10% of the people with shingles, though, that pain goes on and on and on for months and months. And we call that post-herpetic neuralgia. Okay, so it can be really an ordeal. Can someone with shingles pass chickenpox on to somebody else? Well, I asked that wrong. What I was going to ask is, can you give shingles to somebody else? And the answer is no. But if somebody with shingles, maybe say holds a newborn baby that hasn't been vaccinated, they can pass chickenpox on to that unvaccinated individual. Okay, so that can be, kind of dangerous anytime you give it to a newborn baby. I had a student ask me in class once. She said, I work at a nursing home and I never had chickenpox and I've never been vaccinated for it either. Should I get a chickenpox vaccination? I said, yes, you should probably go today because our incidence of shingles gets higher and higher the older we get. And so you're around this population, nursing homes, where they would be more likely to activate, their immunity's waning. And when you get chickenpox as an adult, it's a lot more serious. So age is a factor. You're much more likely to have some nasty complications from chickenpox if you get it as a teenager or adult. Okay, so we'll say contracting chickenpox as a teen or adult is, again, more dangerous. You're going to have more inflammation associated with it, just a higher, more risk of tissue damage, of getting that like pneumonia and encephalitis. Dr. Baxter, some of you may have him for lab. He remembers getting chickenpox when he was like 16 and he got pneumonia as a result of it. So it's dangerous to get when you're older. So that's why, if you haven't had it, then yes, you should be vaccinated to it. What was the other thing? Oh, so the question also may come up, if you never had chickenpox and you were vaccinated to chickenpox, can you ever get shingles? It seems like no. We don't have good data for that because, the vaccine came out in '95. The first people that got it are still in their 20s. You're not likely to get shingles in your 20s, although it's possible. But actually, there's a small risk. Like if you've had the chickenpox vaccine, you can possibly get shingles, but you have a much less likely to get shingles from the vaccine than if you actually had chickenpox. Okay, so kind of an interesting permutations, lots of things there. What is the vaccine, the chickenpox vaccine? It's called Baravax. And this is an attenuated vaccine. That's how, so it's a live vaccine, it's just weakened. So that's why there is a small possibility that it could still reactivate as shingles later in life. Typically, kids will get it when they are somewhere between 12 and 15 months, and then a booster about the time, before they would go to school. And that's good enough protection. We do have a shingles vaccine. So for adults that had chickenpox as kids, to try to keep it from reactivating, it's called Shingrix. And that is a really good vaccine. It has greater than 90% protection. And even if you do get it, it's most likely to be fairly mild. So it's an inactivated virus. This replaced our old vaccine that was called Zostavax, which didn't work as well. It only protected patients maybe 50% of the time. So the newer vaccine, it works much better. We have to give 2 doses, and there is unfortunately, a little higher chances of side effects. So I remember when I had my second dose, like my whole body hurt. Like I remember just laying there and just like, I just feel terrible. I took some Tylenol and then I felt better. So it wasn't that bad, but for me, it was worse than the second COVID shot. I had like all those in the same year. So, all right. So this is recommended for adults that are over 50, just because the chance of reactivation kind of gets more and more the older we get. So encourage your parents or your grandparents to get that, just to avoid shingles. Yeah.
Do you think there's going to be a point where the shingles vaccine will have to be a thing because we're all chickenpox?
That's a good question. Like maybe eventually that will kind of phase out because it's so rare for anybody to get shingles anyway. Yeah, that's a good question. Yep. In terms of when patients do have chickenpox or shingles, what medication do we have? Really just have acyclovir to try to reduce the itching associated with chickenpox and the pain associated with shingles. We don't really have a lot else. Okay. Any other questions about chickenpox, shingles? Okay. All right, good. Let's start our other DNA virus. I started talking about this virus last year because we had the monkeypox, now called Mpox, outbreak. And I realized that really to talk about Mpox, you had to talk about smallpox first because it's so closely related. So the causative agent of smallpox is in the family Poxviridae in the genus Orthopoxvirus. So there are 4 species of viruses in this genus. There's the variola, the vaccinia, what's called cowpox, and monkeypox. All right, so the vaccinia, that is actually the vaccine for smallpox. So it's actually back in Edward Jenner's day, where he scraped some lesions off a milkmaid with cowpox and injected them into other people to protect them from smallpox. He used cowpox. Today, this vaccinia virus, it's kind of a mix of cowpox and horsepox that's kind of mutated over time. So it's kind of, we call its own virus. So it's not really cowpox that we use now. Variola, that's the virus that causes smallpox. So variola, and there's 2 variations of that. Variola major, that causes really, really severe smallpox, has about a 30% mortality. And then variola minor, for some reason around the year 1900, there was some kind of widespread attenuation of that variola major, and the virus just kind of wasn't as deadly, more like a 1% mortality. Okay, so it was much less severe. Kind of the vital signs of that virus. It's another linear double-stranded DNA, kind of like herpes virus. It's enveloped like a lot of viruses are that infect humans. Not all of them are enveloped, but a lot of them are. And it has a very large, complex capsid. So in this picture, you can see the envelope around here, this kind of barbell shape in the center there, that's the capsid. All right, so that's not really, it's not a polyhedron, it's not linear, it's this kind of, it doesn't fit into either one of those, so we call it a complex capsid. And it's a pretty big genome. I think I forgot to say that for herpes virus. Herpes virus has like between 80 and 100 genes. Smallpox has about 200 genes. Okay, so in general, the DNA viruses tend to carry them with them a lot more genes than the RNA viruses. COVID, for instance, has like 7 genes, influenza, 7 or 8 genes. So they're much smaller. Okay. What does smallpox look like? Hopefully, I kind of make you a little bit more appreciative that we don't have to deal with this disease anymore. It was very contagious, often disfiguring. Even if someone survived, they would have scars from all those pock marks. Again, about 30% of the time historically deadly, until much more recently it was a little bit milder. How many people got this? I checked this figure 3 different times because it's so kind of hard to believe, but about 300 million people worldwide were killed just in the 1900s. Okay, so that's why I kept checking that with 300 million people, and the mortality was only about 1%. Like, and we eradicated it in 1980. So that was in the first 80 years of the last century, 300 million people die from this. It's just astonishing. And in the year 1967, we had 15 million cases. Okay, so not deaths, but as late as 1967. I mean, we eradicated it almost 10 years later, and we're still having that many cases. So it's really astonishing. Again, flu-like symptoms, systemic inflammation, especially when someone inhaled the virus, and death was typically by shock. Okay, so much inflammation that just kind of all the organs shut down. Okay, so we started vaccinating in the mid-60s. Must have got on that pretty hard because by 1980 we were able to declare it eradicated. We stopped routine vaccination in 1972, and what time we got here? We're almost out of time, but I want to talk about why we stopped that next time and why you can be glad we don't do that anymore. It was kind of a nasty vaccination process. So we'll pick up from there and finish up with the RNA viruses next time. Have a good day.